these five primary sensation of the test. So five basic test modalities are there. They are sorry, they are sour, salty, sweet, bitter, and umami. But the neurophysiologists have also looking further, and they have found that there are at least 30. But at your level, you should be knowing that there are five basic test modalities are there, and they are sour, salt, sweet, bitter, and umami. And if you look at here, pain, who are highly sensitive to bitter taste. Okay, who are highly sensitive to bitter taste because the poisons, most of the poisons are bitter in taste. So that's what the importance. We have to develop protection against the dangerous toxins in food. So the toxin substance that would ingest, they are mostly the bitter taste. So when the taste is bitter, then we will not desire to take it, isn't it? So that is one advantage. If you look at their different taste modalities like sour taste, they are caused by the acids, by the hydrogen ion concentration. Salty taste is mediated by ionized salts, mainly the sodium ions. And sweet and bitter, they are mediated by different diverse classes of chemicals. Okay, there are different ones. Saccharin, you are already knowing that this saccharin is the sweet in taste, bitter. Okay, there are different. I will show you. And umami taste is mediated by the glutamate. So food containing glutamate maximum, this is umami, that is monosodium glutamate. Monosodium glutamate is having the umami taste. So these are the different chemicals. Okay, these are the different chemicals. They will have NaCl, they have salty taste. Okay, if the hydrochloric acid is also sour in taste. Okay, if it and another is that there are bitter substances are there like caffeine, atropine, cocaine, morphine, strychnine, all these, and quinine. These are the bitter taste substances, and sweet. These are the saccharins, sucrose, fructose, glucose, maltose. Like all these are the substance which is having the bitter, sweet taste. And I have already told you that substance having more amount of glutamate or is having glutamate is having the umami taste. Now there are threshold level is there. If you compare the threshold of different taste, then you can see that for the bitter, the threshold level is very low. Okay, I have also told you the importance for having the low threshold level, or who are having the very sensitive to this taste. So these are the for the different test modalities, we are also having the different threshold, and threshold is minimum for the bitter taste like strychnine and hydrochloride, like that. Okay, now let me discuss on the receptors. Okay, the taste receptors. Taste receptors are actually the modified epithelial cell. Just in my first class or today's class, I told you that olfactory receptors are the part of the olfactory sensory neurons, isn't it? So the olfactory receptors that I have already told you that receptors are present on the cilia. Actually, they are the part of the peripheral portion of the sensory neuron, the sensory olfactory sensory neuron. So there is a difference in between the taste receptor and olfactory receptor. Don't get confused that on that day. The taste receptor are modified epithelial cell, but the olfactory receptors, they are the receptor. They are part of the olfactory sensory neurons that is present on the cilia. Now, talking on that, if you look at here again, this also emphasizes the difference between the difference between receptors of taste and the olfactory taste receptor cells are not neurons but rather modified epithelial cells. If you look at here, this taste receptor snaps onto the sensory neuron that communicates with the CNS. That I will show you. Okay, that this cells, that is the modified epithelial cells, they will form the synapse, and these cells, taste receptor cells, will produce the neurotransmitter or the chemicals. They will release the chemicals, and that chemicals of the neurotransmitter or the substances from the taste receptor cells act on the this neurons, sensory neurons, and they will take the uh, uh, they will produce the action potential on the neuron, and that information ultimately goes to the brain for the detection of the taste sensation. Now, looking at that, this if you look at here in the tongue, we are already knowing that this taste receptor are concentrated in the small but visible projections called papilla. So papilla is there. If you could see on your tongue also, in your mirror, you can see the papilla, the projection you can see. But this projection itself consists of the taste buds. Okay, they consist of the taste buds. Okay, their taste buds are there. There are different taste buds are there, and this is foliate, circumvallate, different types of is there. This is the papilla, you can see it, or the receptors are there. And if you look at here, this seventh cranial nerve that I have already told you in your practical also, anterior to third of the tongue is supplied by the branch of the chordae seventh cranial nerve, whereas the vagus nerve part of the tongue is supplied by the vagus nerve. They will innervate the taste buds, which consist of the taste receptors. Okay. Now, talking on that, if you look at here, then the taste bud is composed of, I have already told you that this modified epithelial cell, the taste buds are the modified epithelial cell, and they consist of supporting cells, that is also known as sustendicular cells, and taste cells itself. This is the epithelial cell, and these get modified, and they are forming this type of taste buds. These are taste buds, and consist of mainly the taste cells. These are taste cells, you can see, and the supporting cells, and these are the supporting cells, and these are the supporting cells. So taste bud consists of these taste cells and the supporting cells, and their cells are the modified epithelial cells. If you look closely over here, you can also see that the synapse, these taste cells are forming, releasing the chemical on these neurons. Okay, and these neurons, so they are forming the synapse over here also. The sensory neurons are there. Okay, they are also innervating. They are innervating the cells, the taste cells. Am I clear?
Yes, sir.
Okay, now let me discuss on that taste transduction. Okay, what is that means? I have already told you this is the signal transduction, and maybe photo transduction or visual transduction. It may be another is smell transduction, and now we are talking on the taste transduction. How the taste or substance that we just intake, that the information ultimately leads to the generation of the electrical phenomenon, and that goes to the brain. Okay, that is known as this taste transduction. Application of this, if you look at here, if we take the taste substances, then I have already told you there are taste cells are there, and taste cell will detect. Okay, they will detect, or they may detect, or directly helps to. Okay, I should figure that at the time it will be clear to you. But right now, if you look at here, then they will act on the receptor. Okay, they will act on the receptor. Or the receptor may be ligand gated, or it may be G protein coupled receptors are there. Different types of receptors are there. So these substances will act on the receptors, or directly this substance can enter also. So depending on that, they will depolarize the taste cells. Okay, they will depolarize the taste cells. I've already told you there are the taste ends themselves, just like in case of sodium, in case of gas plus hydrogen, they themselves enter inside these taste cells. Or in case of this glucose or this sweet, bitter, they will act on the G protein coupled receptor, or different types of receptor are there that I will show you. There are different receptors. Okay, they will be acting on the receptors, and they will may generate the second messenger, and that second messenger leads to the ultimately that will causes the different electrical activity occurring on the test cells, and they have caused the depolarization of the cells. Okay, and that causes this receptor potential, that generation of potential receptor potential, and that causes the influx, cause the opening of the voltage gated calcium channel, and calcium causes the release of the transmitter molecules. Okay, this is the general that you should be knowing. Okay, this is a general mechanism. It may be applied to the different chemicals also, but this is the general, isn't it? You have this is the general. You might read in other system or other mechanism as well when you are taking on the hormonal mechanism as well. So now hormonal mechanism is also some of the hormone they will generate second messenger, or some hormone they will directly interest like that. Okay. Now I will show you the figure, and that will be more clear to you. Now look at here. So figure like the salty and sour. Look at here, salty. I have told you that this testants that directly pass through the ion channels. Okay, the testants that directly pass through the ion channel, just for the testant, this salty and sour means salty means sodium and sour means H plus. Look at here, sodium. The sodium is directly entering inside the cells. Which cells? This is the this this is the test cells, isn't it? So it is entering directly. If we are for the sour, that's H plus, there is also the influx of the H plus to this sodium epithelial sodium channel. Okay, epithelial sodium channel. So this directly this type of testants, this for the sour and so they directly endorse, and they ultimately leads to the opening of voltage gated calcium channel. So this is the voltage gated sodium channel, and the membrane get excited, and that causes the release of the neurotransmitter. Okay, the neurotransmitter in this case is serotonin. Okay, the serotonin is being released, and that will excite this sensory neurons. Look at here, itself, this H plus is also blocking this potassium channel. H plus is also blocking the potassium channel, means the membrane get depolarized. Here also membrane get depolarized. So membrane get depolarized, receptor potential is depolarized. If the receptor potential reaches to the threshold, action potential, okay, in this case, there will be further opening of the calcium channel will be there. Calcium causes the efflux of this basic, or the efflux of the serotonin that is stored in the vesicles. Okay, am I clear? So this ion channel, this is the ion channel, also known as this ligand gated channel, or this ion trophic receptor. So the sodium and this sodium or sour or salt test substance will act on the ligand gated channel or the ion trophic receptor. There will be influx of this. Okay, like this, and there will be the salt, sodium, sour, gas plus. Okay, am I clear?
Yes, sir.
1, 2, 2,
Yes, sir.
3,
Yes, sir.
Yes, sir.
4, 4. Dental, from dental also, please let me know. Hello? Hello? It is not clear to you? Dental? BDS? BDS is not there? Deepika?
It's on, sir.
खै त बोल्दै बोल्दैन बुझेन नि क्या how relax sour taste, but there are other test ends that will activates or they will produce the second messenger inside the cells and they will do their functions. So if you look at here, sour, bitter, and umami taste are triggered by the activation of metabotropic receptor. In previous, I have told you that salt and sour is triggered by the activation of the inotropic receptor. But here, this is the difference, okay? They are activated by the metabotropic receptor. And there, this calcium triggers the ATP release as the transmitter. But here, I have told you that the ATP triggers the serotonin release. Now, these sweets are mediated or detected by the T1R families. Okay, these are the receptors that belongs to they will generate when this test ends act on this receptor. Then there is there is complicated mechanism is there. Okay, that's not using of it like that. They will be activated and they will produce calcium channel and they are similarly test is mediated by sometime in the about the test model. You should be able to write this one. Okay, test modalities. If this is facts, you should state the 5 type of test modalities, and you should write this one and this one as well. Okay, this one as well. So they are these test chains like this and this are mediated by the activation metabotropic receptor, and they will push a second messenger by the activation. Okay, like that. In most of the cases, when this metabotropic receptor are activated, gosteucine is being activated, and that will lead to the production of the second messenger, and that will cause the influx, opening of the voltage calcium channel, and the release of ATP. Now talking on that also, now look at here. Now I have told you that they are being activated by the metabotropic receptor, isn't it? Actually, if you look at here, sweet sour apps use same second messenger. If so, how to differentiate these tastes? They are in the same type of the second messenger. This. So this is the answer. It appears that sweet sensing taste cells do not express receptor for either bitter or umami. If the taste bud is having the receptor for sweet, they will not express for the other taste like bitter or umami. So there will be selective activation of the taste cells, and say there is generation of this action secondary messenger, second messengers like that. Am I clear? Is this clear? It is
Yes.
clear? Is this clear?
Yes, sir.
Okay, fine. Now let me discuss on the how the information ultimately. This is the mechanism. This is the cellular mechanism. Just I have discussed on the cellular mechanism that is occurring on the test cells, isn't it? The test cells or test buds. Now I will discuss on the pathway, how the information that will be ultimately transmitted to the central nervous system. If you look at here, and I have already told you that there is the test buds are there, and depending upon the test, test cells are activated, and they will release the chemical, and they will activate this part of the neuron, that is the synapse is there. So action potential. So action potential is being generated, okay, over here. And if you look at here, the action potential is being generated anterior 2/3, anterior 2/3, posterior 1/3, even from the pharynx. If you look at here, this is the branch of conducting canal, 7th cranial nerve, So the information from the anterior to third is being taken by this first order neuron. This is the peripheral portion. This is the central portion. Okay. This is the ganglia, bipolar cell. This is bipolar cell. So the first order neuron synapse. If you look at here, this is also the first order neuron, and this is the name of the ganglion. This is the name of the ganglion. This is the name of the ganglion for this 10th cranial nerve. 9th cranial nerve and 7th cranial nerve. Okay, geniculate, petrosal and nodus. Okay, depending upon their this cranial nerves, they have got different ganglion. And these first order neurons, okay, this first order neuron, they will synapse over here. They will synapse over they will terminate over this nucleus tractus nucleus solitary tract. Okay, they will be this terminate on this structure. So from here now, from this nucleus of solitary tract, okay, nucleus tractus solitarius, then the second order neuron get origin, and that second order neuron, look at here, the second order neuron terminates over here. That is VPM, ventral postmedial nucleus of thalamus. Okay, VPM, and from there, thalamus, the third order neuron get origin, and they will terminate this third order, this cell body is the axon, and that will terminate on this part, this gustatory cortex. This is present in anterior insula. Okay, this is present in anterior insula, frontal upper column or gustatory cortex. Okay, am I clear? In this way, the information is transmitted from at the level of taste bud ultimately to the insula. Am I clear?
Yes, sir.
Now my assignment for you is try to find it out. What is the Brodmann's area for this gustatory cortex? Anyone knowing? Dungyal is there. Dungyal is there. Deepika, Dungyal sir. Dungyal is there or not? No, Dungyal is not there. Hello, Deepika.
छैन होला सर.
China. Oh, okay, fine. So so these are the paths that you should be knowing. If the question is being asked about this, how draw the pathway for the test signal transmission to the central nervous system, then you have to draw this figure. Okay. If you cannot draw this system, then you have to draw this schematic diagram as well. Okay. You simply draw the arrow like that, and you have to draw this schematic diagram. If you can draw this this type of diagram, that is very good. Otherwise, you have to draw this schematic one. Now, if you look at here, now from this nucleus tractus solitarius, okay, the information is going on this one. So from this nucleus tractus solitarius, the information is also going to the superior salivary nucleus and inferior salivary nucleus. So whenever we test the substance, then they also activates this type of the nucleus, and this you are already knowing, this are for the production of saliva. So this nucleus get activation means they will activate these glands. So whenever we put the substance anything in our mouth, then there will be secretion of the saliva. So this is the mechanism. Okay. So from the tractus solitarius, many information also goes to this nucleus, and this nucleus turns information to this gland. So it's salivary gland, and saliva is the information. Am I clear?
Yes, sir.
Okay, fine. So there is the adaptation is also there. It's the same mechanism for this one also. Now I think the class has been finished. The class has been finished.